Alright, in our previous video, we used the program I created called mybioeditor.py, and we located what we think is a CPG island in our sequence called C3. This is what I have up here on the screen, and as you can see, we searched for C3 and we found here all of the blue highlights are where the C's and G's are. This looks pretty much like a CPG island. We would suspect that there's probably a couple of transcription start sites and promoters in here, but realize that there are multiple types of promoters. Most people are familiar with a data box, but there are actually many different types. And here, if you go to the file that you download for this week's assignment called the Recommended Order of Tasks to Annotate a Gene, you'll find some information about not only CPG islands, but also transcription start sites and promoters. We're going to use this to basically begin the annotation of our sequence. We're going to scroll down here to part 4. You can read parts 1, 2, 3, and 4 and 5. For today, it's just 1 and 4. And we're going to go down here to 4, and we're going to read about broad and sharp promoters. Broad promoters tend to be the promoters that exist, and there are multiple copies of them at the beginning of a gene. So that means that there are multiple places that transcription can begin in association with that particular protein coding sequence, or perhaps even non-protein coding sequence for that matter. And here we have a sharp promoter, like a TATA box, where there's typically only one start site. So if you find a TATA here, you're going to say this is the only start site. It's typically the way you would state that. All right. Now notice it says they're typically not found in high GC regions. That means you're probably not going to find a TATA. You might. You do need to look for it though. All right. And then we come down here and we have intermediate, which is your GC box. GC is actually the most common promoter. Most people, the better known is TATA, but the most common, at least in humans, is the GC box. So let's go ahead and let's search for a GC box. The GC box is 3 G's, a C, and 2 more G's. So 3GC, 2G, if you want to kind of say it the way I kind of remember it, GGG, C, GG, and this is going to be in green. All right, here are our locations for those. Let's take a look where those show up in our sequence. Well, look at that. They show up right in the middle of our CPG island. This is an indicator, at least to me, that we have multiple transcription start sites for this particular gene. You would need to go through here, find out how far a CPG island usually is from the transcription start site, and come up with a location that you think is the most likely location for the transcription start site. Now, there are some hints. You will need to read my description here, like where do you normally find a transcription start site relative to your promoter? What sequence is it? For instance, I tell you here that CA in mammals is more typical than other sequences as your transcription start sites. You might look for a CA. Here it says that GC boxes are at -200 to +50. Oops, that doesn't tell us too much, but it does tell you optimal, it's at -10. Now what does all that mean? That means that if it's a minus, it's upstream. It's before the transcription start site. If it's a plus, it's after the transcription start site. Not all promoters are before the location that the polymerase bonds. Okay, there are lots of things going in there, lots of DNA folding around. There might be some RNAs involved, who knows, and probably dozens of proteins. The idea here is to find where you think the optimal place is, or as close as you can get, because you do this and then you go into the laboratory and you do your experiments and see if it matches up with your data analysis here that we have. You might go and do some type of say footprinting, where you find out where the the protein binds to the transcription start site, and find out if you're correct. All right, that costs money. This will give you a kind of a hint as where to look for it. All right, now we look up here. When you do a DPE, you have a W and a Y. What does that mean? Well, for that, you need to go to another file that's in this week's folder, and this is going to be the IUPAC symbols, and this is going to tell you things like, oh, we need to not look for a G or a C. We need to look for, say, a G or A or a C. If it's a V, we're looking for W's and Y's right here. W is an adenine or a thymine, and a Y is a thymine or a cytosine. So once you get a feel for that, all right, you can go in and search for multiple characters. Well, the problem is, how do you do that in this search? Well, if I want to search for a or a T, for instance, you type that in by using brackets. So maybe I'm looking for G, 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 and then maybe I'm okay with an A or a T. I put that within brackets. And then maybe the next single letter I'm looking for can be a G or a C. I put that in brackets. And then maybe I want 3 C's. I want to find this particular 8-letter sequence. 3 on either side, G's, C's, and then an A or a T at one point and a G and a C next to it. And we'll see if this sequence exists in ours. It does. There are 2 of them. And let's see where those are. Here. They're way up here in the beginning. They don't have anything to do with it. So if this was a promoter, maybe it's a promoter for a different gene. There are 2 genes in this sequence. I'll give you a hint. And 2 known genes. Maybe there are more. Maybe you'll find an additional one that we don't know about by analyzing it. But the idea here is these, all right, the red ones, are outside of the parameter that we're looking for. We're looking around this CPG island. This is the hint that's telling us that this is interesting. This is what I'm interested in. So you get something like this. I'm going to tell you to ignore it for our assignment. We want to find the transcription start site down here in this region. All right. You want to find the promoters, and you want to find the possible transcription start sites. And that is week 4's assignment.